Hello and welcome to Apologetics 101. I'm your host, Brian, and today we are starting our very first episode of Are We Apes? Of the Are We Apes series. Are We Apes? We're going to be asking the question, are we apes and are we related to apes? Which is what is being claimed by the evolutionary community, because that's ultimately what they claim is that human beings are related to apes and chimps and that we share a common ancestor with them. Um, Usually estimated between 6-7 million years ago, at least as far as when they think the split occurred and that sort of thing. And so we're going to be dealing with that in the series. Today we are interviewing Dr. Georgia Purdom on related to apes, and we're asking the question of genetic differences between humans and apes. And obviously we're going to zero in and deal with a lot of different subject matters as the series goes forward. But today we are going to be doing genetic differences, you know, things involving epigenetics and things like that, and certain questions that we have for Dr. Georgia Purdom, who I've heard is an expert on the subject matter. In fact, everyone pretty much says she's the person to go to when discussing this topic. And so today we got Dr. Georgia Purdom on, and well, without further ado, let's dive in.
Hello, my gentle and, of course, very modern apes.
Sure, I'd like to kiss you goodbye.
All right, but you're so ugly.
Human beings are not just related to apes, but actually aren't.
This is a primate, and so is this, and so is this, and so is this. Hey, that's me.
We are still hominids. We're still primates, mammals, tetrapod.
Hello. Welcome to Apologetics 101. I'm here today with Dr. Georgia Purdom to discuss if chimps and humans are related, concerning mostly, primarily genetic differences between humans and chimps. Am I coming in good, Dr. Purdom?
Yep, loud and clear.
Awesome. All right. If you can, just kind of give, I know you've been on this channel before, but just kind of give a brief introduction to yourself and then we'll jump right in.
Sure. So my name is Georgia Purdom, and I have a PhD in molecular genetics from Ohio State University. And I've been at Answers in Genesis 18 years now. I'm the Vice President of Educational Content and also now the Director of Research and the Editor-in-Chief of Answers Research Journal, which is our technical peer-reviewed journal.
All right. Awesome. All right. You're kind of like considered an expert on the subject, ain't you? On chimps and human differences.
I've
Everyone,
looked at it a lot. I've definitely read a lot about it. That's for sure.
right? All right. The first question I wanted to ask you, there is a specific issue that's brought up every time. I kind of basically just ran this issue on my channel because I had recently a series called the Tompkins Controversy. So that this has been an extensive thing, and it's of course it's something that's blown up on YouTube a lot. So on this 96 to 98% similarity between humans and chimps. Could you address that and discuss the whether that is the case or not, and what issues that might involve?
Yeah, so one of the things that Tompkins research is really good in this particular area. I mean, does a really good job with addressing a lot of that, because he is a genomicist by training. I mean, he deals with genomes and comparing them and understanding them. And so, um, so what so what he's done is not just done his own research and comparing the genomes and giving those kinds of numbers, but also done a lot of research in, well, how were the comparisons done by the evolutionists? And one of the things that people don't realize is when you hear that 96 to like 98% similarity, they're really only looking at one type of difference. And it's basically what they call substitutions in aligned regions. And so there's, if you ask a computer to compare human and chimp DNA, because they're massive, right? There are 3 billion bases, so 3 billion base pairs. So it's a lot. There will be areas that are similar in humans and chimps, which just like there'll be areas that are similar in humans and dogs and humans and bananas. Okay. You're going to have some similarities because we do come from the same uh, creator God. And so, and banana cells have to do things that human cells have to do as well. So they are doing similar things. So that just makes sense from that perspective. So in those areas which are very similar, okay, in sequence, there will be areas where you have, let's say, human DNA is a T, but chimp DNA is a G there, okay, because we have, DNA is 4 bases, A, T, C, and G. So those areas where you have DNA, but it's different in the aligned regions, or like, where it's a T versus a G or something like that, that's what they're basically counting when you see the 96 to 98% similarity. Now, I always say, here's the thing. Do we know that those are the only differences that make a difference between humans and chimps? And the answer to that is no, we don't know that. No one's ever done an experiment to show, oh, well, these are in these regions and this is why chimps and humans are different. No one's ever done that. The reason that number, you might say, I mean, my logical next question would be, well, why are you not counting all the other differences? Because there's lots of others. There's areas that are not aligned at all between human and chimp. They're so different that the computer won't line them up. They're unaligned regions. There's even if you look at aligned regions, there's areas where there's gaps where you have human DNA but no chimp DNA, or chimp DNA but no human DNA. Don't count those. None of those are counted. There are legitimate differences between human and chimp, but they're not counted. And I think the biggest reason why they're not counted is because if you count all those differences, it's going to lead to a much larger evolutionary time frame that you have, to be able to get the differences between humans and chimps, because supposedly they had a common ancestor 6 million years ago. So in order to get that many differences, you're going to have a lot more time. Or you're going to have to say a lot of those differences don't matter. Well, they're in junk DNA. Well, as we study more and more DNA, we know there's no such thing as junk. So that argument's really hard to put any weight to. But you could reasonably say that many mutations, substitutions in aligned regions could occur, that 1% difference or so, in 6 million years. Now, I always say that's the best case scenario, right? The reality is that even if you could get those differences, that 1% or 2% difference, mutations, changes in DNA do not do what evolutionists need them to do. They don't add genetic information. They don't make an organism change from one thing to another. Instead, they degrade and they deform and they kill things. They don't give you anything that's good. So even if you could get those, even if you got those changes, or even if somehow you could get all these changes that you see, the differences, you're not going to get anything that's living. You're not going to get anything that's good, because that's just not what mutations do. So there really is not a basis for saying, the 96 to 98% similarity. And Dr. Tompkins' work with his all of his research that he's done seems to fall around 85% similarity between the two. And that's just looking at more the linear sequence of the DNA, like, base for base kind of thing. The reality is, I always say DNA is a 3-dimensional structure. And so there's literally, as we continue to study it, layer upon layer upon layer of information. You've got things like epigenetics, you've got that. And we see there's differences between humans and chimps there too. So it's more than just that linear sequence that matters for talking about the genetic differences between humans and chimps. There's lots of other layers, and the more that we study those, the more differences that we see.
Yeah, I was talking to Dr. Tompkins about that the other day because he was talking about layers of information and stuff like that. there was a critic that was trying to debate me over what information is, and they were using Shannon's theory of information, which really doesn't tell us anything about the meaning of it's about statistical weighting of information and things like that. Um,
Mhm.
but in any case, all right. Yeah, I when I looked into it, there were like these algorithms that are used to make these comparisons, BLAST, LASSI, NUCMER, and things like that. They're looking for similarities. They're looking for sameness in the genome. They're not really looking for differences per se. And
Mhm.
so there's a lot. If there's like a large region of the genome that's just not comparable, that's not alignable, it's not going to pick those up because it's only looking for similarities, not differences. And on substitutions, you mentioned substitutions. Would you say that that is assuming common ancestry? Like if they see a sequence and you're supposed to put a G, it's a G there, in chimps, and it's a T there in humans, then they assume that at some given point there's a mutation that changed that, and so they
Yeah.
just kind of assume common ancestry.
Yeah. I mean, even initially, and I don't know if Dr. Tompkins talked about this, but initially we had a very humanized chimp genome. So the way the chimp genome was originally assembled was based off the human um, as a template, basically.
Yeah, it's
It's
a scaffold. Yeah.
yeah, just last
Okay.
year we got the first de novo chimp genome. Yeah.
Yeah. I think they've just about got that complete now. But at one time that they had I think it's called shotgun sequencing, where they try
Hmm.
to use the human genome as a scaffold because they assume we're related to chimp, so that we're the closest living relative, and so that's why they used that, and then they compared it and
Yeah.
reassembled it.
Yeah.
Um, there is an interesting thing that you raised up. I got a book here, uh, Glass House.
Oh,
I had,
uh-huh.
yeah, I already had that book when I invited you on. But I was very interested in what you said about genetic markers. You also mentioned that just a second ago.
Yeah.
What are epigenetic markers?
Yeah, epigenetics is it's a fairly new field of research with genetics, maybe in the last 20, 25 years or so. But basically they're chemical groups that are added to certain bases in DNA. So for example, methyl groups like CH3, carbon, and three hydrogen can be added to cytosine, which is a base in DNA. You can have acetyl groups added. And so they're kind of like they're tags that are added to the DNA. They can also be added to histone tails. So histones are the proteins that the DNA wraps around. And so they kind of have a tail-like structure that those epigenetic markers can be added to as well. And that probably has to do a lot with controlling the unraveling of the DNA and whether it gets transcribed and translated and made into like a protein or can be accessed basically. And it probably has a lot to do we think these methyl groups, acetyl groups, there's like I think there might be like 10 or 12 different categories of chemical groups that are added. We think they have a lot to do with regulating the expression of the DNA. Because if you think about it, it's so much more than just producing proteins. It's when, where, why, how much, under what condition, excuse me, that you have to think about all of that. And all of that has to be controlled. I mean, if you compare across the board, let's say humans to rats, to cats, to dogs, to chimps, we're going to have a lot of the same genes. But we're obviously very different organisms. So I think a lot of that epigenetics is responsible for controlling, those things and making some of those differences, at least between the organisms on a biological level. And uh, I like to think of them kind of as they're kind of like punctuation, in sentences. So if you think about like if I could read this two different ways, the same words, you'll see the same words, but when you hear me speak them, you can tell there's different punctuation. So I could say a woman without her man is nothing. Okay. Or I could say a woman without her man is nothing. Now they're the same words, but you put different emphases when you talk because there's different punctuation and you have completely different meaning. And so It's very similar with epigenetics. You've got maybe the same sequences, but you have different markers, and so it's read differently and or expressed differently, the DNA. And they think too, like if you think about identical twins, they have the same DNA sequence, same. There's nothing different about it. But sometimes one twin will get a disease that the other twin will not. And why is that? I mean, it can't be genetics, right? You think their genetics are the same, but are they? And they've even done studies to show that the epigenetic markers of identical twins, like shortly after they're born, it's very, very similar. But as they get older and maybe they don't live in the same area, they have different experiences, they eat different foods, their epigenetic markers start to get very different over time because they're just, again, it's like and that's how maybe why one has cancer and one doesn't, because it's just a difference in how that DNA is expressed. And so and when we look at that, I had mentioned earlier about the difference between humans and chimps on that. Dr. Tompkins had cited a study that was done on white blood cells, which are the neutrophils. That's a type of white blood cells, the most similar between cell type between humans and chimps. And he had said there was a paper that basically said the methylation differences, which is epigenetic differences, there's methylation differences in 1,500 different regions of the human DNA compared to the chimp DNA when it comes to their white blood cells. And so it is very, it can be, so it's just this other layer of information that's different. So if you think about evolving, humans and chimps coming from the same ancestor, well, you've got to do a lot more than just, the genetic changes, the DNA changes. You also have to make the epigenetic changes, which is a whole nother layer of information. And so, yeah, how do you do that? How do you account for that by evolution?
Right. And my next question is very related to that. Do humans and chimps or apes share epigenetic markers, or are they different? You
So
addressed that a little bit.
yeah, so they are different, like the white blood cells I just talked about. There was another study where they looked at human gene promoters. So promoters are regions of DNA that promote the expression of different genes, and they're less methylated in humans, certain gene in the brain. Certain genes are less methylated, which if they're less methylated, it usually means they're more active. So they have more proteins being produced from those genes. If they're more methylated, or sorry, less methylated than the chimps on those same types of genes. So there was like, I think over 1,000 genes in the brain between humans and chimps that showed significantly different methylation patterns. So significantly different epigenetics. So those genes are producing a lot more proteins in humans than they are chimps. So it's just another difference because we're not related.
Right.
We're created.
But if we were related, you would expect the epigenetic markers to be the same. If we just
You
kind
would
of
expect
branched
them,
off.
yeah, maybe not identical because there has been 6 million years, they would say, since we evolved from a common ancestor, but I would expect them to be a lot more similar.
Yeah. Um, all right. Did two human chromosomes fuse together? That's another thing that I've actually discussed on my channel. But
Yep.
sometimes chromosome two fusion is brought up. All humans have 46 chromosomes and apes have 48. And they say, well, two of the eight chromosomes just kind of fuse together to form one of our chromosomes in common ancestry. Is there problems associated with that?
Oh, big ones. That's one of my favorite things to talk about because it's one of those favorite evidences that people have. And I like to say, you have to look at the sequence. You really have to look at the sequence. And so if two chromosomes has fused together, there should be a lot of what we call telomeric repeats. So telomeres are sequences found on the ends of chromosomes. And they're typically just repetitive sequences over and over again. And without going into a big long explanation, there's a reason that they exist that way. And basically every time DNA makes a copy of itself in replicating cells, it gets shorter. And so those protect the stuff in between the telomeres. So they're on the ends and they're protecting the stuff in between. So if two chromosomes have fused end to end, there should be a lot of those telomeres. There should be hundreds of thousands of them in the middle of human chromosome 2. So they would say chimp chromosomes 12 and 13 fuse to form human chromosome 2. And that this happened after they split off from the common ancestor, because obviously chimps have 48 and we have 46. So it happened some point after that break off. So the problem with that is if you look at that region in the middle there, there's just not that many repetitive sequences. You would expect a lot.
700 and something minute of bases long.
It's a lot smaller than what you would expect. So that's one thing. Secondly, Dr. Tompkins has done some research on what that region, and he says it's something called interstitial telomeric repeats, which is like a really long, term. And he says they have functional roles. They're not junk DNA. They're not telomeres. They just look a little bit like telomeres, but they are actually found throughout human chromosomes, not specific to human chromosome 2, and they have function. The other thing is there's a gene there. So where the telomeres meet and where the telomeres supposedly meet and come together, there's a gene. And genes are never in telomeres because they get shorter every time the DNA makes a copy of itself. And so you can't have that. You don't want genes to disappear. So that would be a problem. So, and we know it's an active gene. And the same is true, like every chromosome has one centromere. So they say human chromosome 2 has 2. One of them's inactive, but it still looks like a centromere. But again, I always say it's same song, second verse. So there's not enough repeats there. Centromeres have a lot of repetitive sequences, and there's just not enough there to justify a centromere. The sequences in the supposed inactive centromere are found throughout human chromosomes. They're not specific to human chromosome 2. And number 3, there's a gene there, and genes are never in centromeres. Centromeres are usually found towards the middle of the chromosome, and they have to do with allowing certain proteins to be there to break the chromosomes apart when the cell is replicating itself and the DNA is replicating. And you just don't find it. So I say and that's just some, but if you look at the sequences, you cannot justify that there's been telomeres or a centromere there. And I'd like to add that telomeres are supposed to prevent chromosomes from sticking together. That's kind of one of their jobs. So like, I don't understand how they're involved in an end-to-end fusion. That's a problem.
Um, uh, I wish we had more time to discuss because you actually do discuss this in your book a lot, or in the chapter that you wrote in that book. Uh, on the some people try to argue that that's a pseudogene, but like you said, that's highly functional. Now I have full videos on this on my channel, so we don't have to go too much into that because I know we're limited on time. But it is that gene has been found to be very functional, and that sort of thing. And like I said, I've got other interviews on this channel specifically on chromosome 2 fusion. But you would say that that's highly functional and it's not a pseudogene
Right.
as they often argue.
Right. Yep.
All right. Does primates and humans, this one I've heard claimed on the internet as well. Does primates and humans share a common deficiency for producing vitamin C? This is also another one you also deal in that chapter I was reading just before the interview. So,
Yeah. Yeah, so they talk about this, it's called the GULO, Gulo for short, because it has a really long name otherwise. So GULO is a gene necessary for making vitamin C. And so primates don't have it, humans don't have it, most mammals don't have it actually. Rats have it for some strange reason, but they can produce vitamin C. But so what's really interesting is if you look at the GULO genes, and they would call them pseudogenes. So they would say they look like we used to have that ability and we've lost that ability. But they want to try to argue, well, the same mutations that you see in chimps, you see in humans, but that's not really true. They're and Dr. Tompkins refers to this as taxonomically restricted gene degradation. So in other words, you get, yes, it does look like a gene that used to be functional and it's not functional anymore, but it's restricted. The degradation that's occurred is really restricted to a particular species. So the human GULO pseudogene, it's 84% similar to chimps, but 87% similar to gorillas, which is a problem. Because we should be, if we have a common ancestor with the chimps, 6 million years ago, our pseudogene should be more similar to theirs than it is to gorillas. And so that's a little strange in that sense. And so that's a problem because they would never say that we're more related to gorillas than chimps. So that in and of itself is an issue. So, and again, overall, what we see as far as similarity goes to chimps is about 85%. And that's what you're seeing even within this particular gulo pseudogene. They're still pretty different overall. There's only an 84% similarity. So I just don't think it has any, again, and we don't know 100% if there's mutational hotspots or things like that, where sometimes mutations tend to occur more often than in other spots. But it just means that, again, maybe we've had the ability to produce vitamin C. Primates had that ability too, and we've just lost it over time. But it doesn't. But there's lots of other mammals that don't make vitamin C either, and no one tries to make the argument. So with them and us, so yeah. That just doesn't seem to hold true.
Um, I know that in your book you said that we're like 87% similar between those in gorilla, where it was
Hmm.
84% similar between us and chimps.
Hmm.
Um, Now, some people try to argue that it's broken in the same place between chimps
Hmm.
and humans. How do you?
Well, they're just not. I mean, you can look at the sequence and see that. So again, there's only the genes are the fact that it's only 84% similar between humans and chimps means that there's lots of places that are different. So if they supposedly we had a common ancestor 6 million years ago that we inherited this from, you would expect there to be in that amount of time, there should be a lot more, a lot, we should not be quite, it just doesn't support that idea, basically, that we have that common ancestor 6 million years ago,
Right.
because they're so
Um,
different.
okay. What does the Bible tell us about human and ape distinctions?
Well, it doesn't mention apes specifically. Only humans are mentioned and in scripture. And when we read in Genesis 1, we see that God created animals according to their kinds. And then God created humans. And so the kinds that God created, the animal kinds, would include the apes, the ape kind. And so chimps are a part of that, and other organisms, would be a part of that, bonobos, things like that. And we know that humans were created separate and different from the animals because they're the ones made in the image of God, and they're told to have dominion over the animal. So we know that we're not related to them in that sense because we're created separate and different from them, and we're not just animals because we're told to have dominion over them, and because we have the image of God. So we are distinct and separate creations, and the evidence supports that.
The evidence confirms it. All right. Uh, in all your research on this, I was wondering if you can give kind of like a final word on it. What is your impression from just looking at the genetics and seeing these kinds of differences arise and things that we would expect genetically, with the epigenetic genetic markers and so forth that that we would expect if we were related to apes and that we're just not seeing. And so what is your final thoughts on that?
Yeah, I think, again, the more and more that we study humans and chimps and, people try to make the argument that we're related, the evidence does not bear it out. I mean, first and foremost, we know what God's word says. But if we look at the evidence as well, we see that it confirms what God's word says. And so it doesn't matter. I mean, over and over again, we see these arguments about pseudogenes and chromosome fusions and, similarity in sequence and epigenetics and all of these areas. But the more that we study those, the more differences that we see, the more we have an understanding of the DNA, which is incredibly complex. The more uniqueness that we see between them and us, and between us and all the other organisms as well. Because God, and again, we expect to see some similarities. I mean, that's just, we come from a common designer God. I can tell if I look at a series of paintings and somebody says which ones are by Monet, I'll be able to pick those out because Monet has a particular style, and I'll probably be able to pick those out of that. I'm not some great, art connoisseur, but I can probably do that. And so the same is true for what God has created. I mean, our bodies, we're both mammals, so our bodies, of course, are going to do some of the same types of things, but they're not going to be identical because, again, we're created separate and different into different kinds. And we do have, I would argue, the humankind, which is separate from the ape kind, and because God created us separately and differently. So I think it's neat. I love being a creation scientist, I always say, because the evidence confirms what God's word says. It doesn't go against it, which is definitely what it's doing for the evolutionary view.
Right. Absolutely. Um, so thank you again, Dr. Purdom. I appreciate it. I know you got
Yeah.
church later, so good luck. Um, I hope everything works out okay. And thank you for joining us on there.
Yep. Thank you for having me.
All right, no problem.
See ya.
Appreciate it. Bye. Thank you for joining me here on Apologetics 101 for the first episode of Are We Apes series. We got a lot more in store. I'm hoping to get a lot more interviews together and zero in on the subject matter. Are we related to apes? Are we related to chips? Are we should we be labeled or classified as apes as evolutionists like to do? And so I'm hoping to get together a good cast of interviews ahead, and hopefully we'll dig into some of the fossil evidence as well, and discuss that as the series progresses. Thank you for joining me on here for part one with Dr. Georgia Purdom. It was a great discussion. I would love to try to zero in some of these issues and stuff like that, but we'll see as the series progresses. And you tell me what you think in the comments. Don't forget to like the video, give the video a thumbs up. That helps the YouTube algorithm and gets this information out to more people. YouTube will help promote it more if the more thumbs up you give it. So also share the content on your community pages and on Facebook and whatever you want to share it. Get this information out there to more people, and hopefully they'll join us as well on this channel. And this channel is growing. It's been growing. It's still growing. And I love every supporter that I have. I thank you so much for supporting me and coming out here and watching the videos. And I enjoy dialoguing with you, and even some of the critics. Some of the critics can be kind of clingy. I still enjoy them on my channel. I enjoy every one of y'all on my channel. Thank you for coming out and watching, and hopefully we have more in the series. And of course, don't forget to subscribe to my channel. If you're new to my channel, don't forget to subscribe. Like I said, like the video, give it a thumbs up. Don't forget to drop a comment below. Don't forget to hit that notification bell. All of these things kind of help the channel and be able to get this information out here to more people. And I hope to see you soon. And we've got exciting lineup ahead. I'm hoping to get Dr. Carl Werner back on the channel. He's kind of booked up a little bit, but he is going to come back later on. We're going to probably schedule something maybe in March of next year. He says he's booked up until that point, and so we're gonna go. We're gonna have to play it by ear a lot with Dr. Carl Werner, but he is coming back. And I'm hoping to have Dr. Gabriella Haynes back on here as well. Talk a little bit more about what's called cladistics and how they're reclassifying these things. I'm hoping to see if I can get her on the series for that reason as well. She said she would come back, and I enjoyed our conversation last time, and a lot of other people enjoyed our conversation. And also, I have a live interview with Jennifer Niza toward the end of this month via StreamYard. Uh, That's going to be the very first live interview I've ever done on this channel, so don't miss that. And I'll see you soon. Thank you.